Hi, this is a seminar dedicated to enhanced rapid NGS tools for viral research. I am Cris Kinross. I work in global market development for the NGS on Illumina platform program at Thermo Fisher Scientific, and thank you. I appreciate you joining us today. So we're going to spend our focus today talking about viral research and some tools that are now available to make viral research more accurate and a little bit faster. Everything we're going to talk about today falls squarely within the mission of Thermo Fisher Scientific. Our mission is to help our customers, to help you make the world healthier, cleaner, and safer. And in the middle of a pandemic and insecurity in terms of safety, there's no better time to focus on this mission. So everything we're going to talk about today falls squarely within why we exist as a company and where we're focused to help you in your work. So why NGS? Well, there's a lot of ways to tackle viral research, and they're very complementary. And so NGS certainly isn't the only technology that would be used for viral research. But one of the benefits of NGS is that it is very, very fast and doesn't require prior hypothesis or prior knowledge. If we take an example from the SARS-CoV-2 pandemic that we're all living through right now, and we look at Lagos, on February 27th, the first case was confirmed. By the 1st, it had been sent for sequencing. And by the 4th, it had been published. So the genome was published very rapidly within days of the first infection. This is the benefit of NGS. They were able to move very fast. And so everything we're going to talk about today needs to be date and time stamped. This presentation is being given in June of 2020. The situation could possibly look very different by November of 2020 or even July of 2020. And so we will be very careful throughout this conversation to tell you exactly when the data was collected to help you understand that it's a snapshot in time. What doesn't change is that the approach for viral research is pretty consistent. 3 phases. Phase 1 happens very fast. It's the identification phase. And this is when we try to figure out what is it that's making us sick. Does it have a name already? If it has a name, has it mutated? Does it not have a name? Do we need to give it a name and assemble a novel genome? This typically happens very quickly, and this phase was really completed by about the end of December, early January, depending on which study you follow. We then move collectively as a scientific community into a positive feedback loop of researching potential vaccines and therapeutics for this disease, as well as monitoring it, just to keep track of the new strains that are emerging and make sure that the work being done on vaccine and therapeutic development is not going to be negatively impacted or changed because of new strains of the virus that are emerging. So what are we going to talk about today? Well, today we're actually going to talk about a new portfolio from Thermo Fisher Scientific called Colibri. And it's worth thinking for a moment about why now? Why didn't we do this 10 years ago? So Thermo Fisher Scientific has been in conversations with NGS labs around the world and taking a close look at the technology that was offered years ago and more recently, we wanted to wait until we could offer something better and fix some of the problems that are still there with older technology. One of the challenges with the older library prep kits is that after a day or two at the bench, working with clear liquids and clear tubes, you run a bioanalyzer trace where you sequence it and you find out, did you actually generate a library? This is a terrible way to approach precious samples. You really don't want to wait that long to find out if you're going to get usable information out of it or not. There's got to be a better way. And so one of the first things we did was we took a hard look at that question is how can we improve the success rate of samples and make sure that you get feedback at every step that things are going as intended. And so how we've done this is that we've introduced unique tracking dyes into each reagent in the Colibri portfolio. You can see a visualization here where you can see 2 reagents being added together and then thoroughly mixed. So Colibri is a made-up word. It translates loosely into 2 Latin words meaning information and full of color. Specifically says these are libraries full of color, information full of color. So anytime you see a part of the Colibri portfolio, you'll know that it has these tracking dyes. So why is this important? Well, these tracking dyes serve a couple of very important pieces of information. The first is for your robot. You need to know that each channel on your multi-channel pipetter is actually dispensing and repipetting as intended. If you've got 96 samples or more, you don't want to wait until the end in QC to find out that one of your tips was actually not seated firmly and the library is gone. You'll be able to tell in the first step, in that first color change mixing step, that everything's fine. The second area where this is particularly helpful is when training new employees. You bring a scientist into the lab to help during surge capacity or as part of a training exercise, and now that scientist can see at each step that things are happening as intended, because that color change only happens when the reagents are mixed thoroughly. The third area where this helps is to avoid over-mixing your samples. If you're working in clear liquids with clear tubes, you might go back and repipette and remix it 2, maybe 3, maybe even 4 times to make sure that they're mixed thoroughly. Doing that, you are further fragmenting your sample, and you might even be damaging some of the enzymes inside, depending on the step that you're working on. So it's important to mix thoroughly, but you don't want to overmix it and introduce damage to your sample that wasn't there when you began. And so these reagents will change color. When mixing is complete, you're good, you can keep going. One of the other areas that we've heard quite a bit of feedback from different labs is that there's an incredible amount of time being spent on sourcing, and this really falls into 2 categories. One is just gathering the materials needed to generate libraries. So you get indexed adapters from one vendor, you get your reagents from a second, you probably get your purification, your cleanup beads from a third. You could even have a 4th vendor involved for plastics and other miscellaneous equipment. This is kind of a waste of the time. It would be much more efficient to just have one catalog number and get everything that you need, and know that those components were intended to be used together and that they were optimized to work together rather than trying to hodgepodge together different reagents. And so what we've done with the Colibri portfolio is that one catalog number will give you everything that you need. You get indexed adapters with unique dual indexes for Illumina platforms. You get a PCR master mix if you want something with PCR in it. There are PCR-free versions available for whole genome sequencing if you prefer that. You also get the cleanup beads. You don't need to go look for your ampere. Just pull the bottle out of the kit and you're good. There's even spare accessories such as seals. So today, in the context of viral research, we are going to focus on 2 primary categories of library prep methods. We're going to talk about whole genome sequencing, and this is where we sequence the host, typically us, but other animals as well. Also, if you choose to reverse transcribe the SARS-CoV-2 RNA virus into cDNA, you would use this approach after reverse transcription. The second category is RNA sequencing. We're going to talk about the differences between 3 different methods of whole-transcriptome sequencing, mRNA sequencing, and 3' mRNA sequencing, and talk about the trade-offs between them and when they might be appropriate for different areas of viral research. 2 other categories within the portfolio. We'll touch briefly on library quantification at the end, but there's also a 4th category of library amplification, and this is when you have very small amounts of input, you generate your library, and then you need to amplify your library in order to have enough to perform QC and sequencing while keeping some for archival purposes. The unique feature about the amplification option is that it doesn't introduce any GC bias. It amplifies GC and AT equally well, and so it's a way to amplify other technology, other kits, and generate more library. Before we dive into how to do this, let's take a moment and think about what we're actually doing. Why are we doing this? Well, within the context of vaccine development, it's a very detailed, fairly long process. It can last anywhere from 18 months to decades, depending on the particular virus under question. Today, we're going to focus on the discovery phase, and specifically, we're going to focus on the viral biology part of the discovery phase. Thermo Fisher Scientific can help with each of the stages in vaccine development, all the way through preclinical development and clinical development. And so if you'd like to have a conversation about antigen design and discovery or clinical trials, how to manufacture at scale, please give us a call and we will help you with those as well. Within vaccine development, some areas of the genome and some areas of the NGS data are going to be looked at a little bit more closely than others. And that's because they're just simply more relevant for vaccine development. One area that the scientific community has been spending quite a bit of time on is the 12-base insertion of the ACE2 protein within the spike protein. And so the reason for this is that that 12-base insertion appears to be the difference between a year ago when we were all flying around and attending weddings and now where those activities require careful thought or frankly should be avoided at all, depending on where you live. And the reason is those 12 bases are the difference between this particular virus being able to enter our lungs or not. Within the phases of viral surveillance and epidemiology, we're asking two questions. The first question is, are new viral strains emerging? This virus mutating? And the answer is yes, of course, all viruses mutate. But more importantly, how quickly and in what way, and what is the impact of those mutations? And so you can see a visualization here on the left from nextstrain.org, who's done a really good job of collecting genomes from SARS-CoV-2 from around the world and visualizing the emergence of new strains. So then the second question being asked in this particular step is, okay, so there's a new strain. What genetic mutations have occurred that account for that strain? And so this allows us to get to very detailed single base pair resolution without any prior assumptions or hypotheses to study these new viruses. We're also, as a scientific community, monitoring where these strains are, where they're going, and where they've come from. At this point in time in June of 2020, this is particularly important in the United States, where it's currently believed that there's one strain of the virus that infected the East Coast of the United States and a different strain that infected the West Coast and may have quite a bit of impact in terms of outcomes for those two groups of people, in terms of how many people became sick and how ill they became. This is truly team science. So one example of a collaborative initiative that is going on is the COVID-19 Host Genetics Initiative. Now, the screenshot shown here of members of this initiative was taken in April. What I would encourage you to do is use this URL at the bottom, which is covid19hg.org/partners, and you can see a full list with map of everybody involved. What's important here is that the scientific community, regardless of background, previous focus area, or politics, has rapidly come together to tackle this pandemic and to work together. And I'm so proud of this community because these are groups that previously might have competed for funding with each other. All of that is gone. Everybody is working together. This is truly a collaborative experience. So that's what we're doing and why we're doing it. Let's talk about how. How are we going to do this? Well, today, let's start with whole genome sequencing. Now, whole genome sequencing in the context of SARS-CoV-2 could be done in two possible scenarios, or two primary possible scenarios. The first is to understand genetic differences among us as people that might account for susceptibility to infection by the virus itself, as well as different strains of the virus. The other is that some labs are reverse transcribing this RNA virus into cDNA, where it's a little bit safer and more stable, and then going into whole genome sequencing. So what we're going to talk about here is applicable to either of those two scenarios. And what's different about the Colibri kits is several things. The first is that there is a PCR-free option available. So particularly if you're going to sequence human samples where DNA is a slightly more abundant, PCR-free might be a good way to go. It saves time and also reduces any possible risk of bias that could be introduced by PCR. But there's also an amplification version. And so the amplification version is really good if you have minimal amounts of samples. And it includes that library amplification module that we spoke about earlier in this conversation, where it amplifies AT and GC equally well. And so it reduces quite a bit of bias that you would have seen with older library prep technology. Is that we wanted to find out with this new library amplification module, what is the impact between PCR and not PCR? Is there an impact? And so what we did was we looked at two extremes. We looked at 1 nanogram of DNA that was amplified in 12 cycles of PCR and compared it to 1,000 nanograms of DNA that was not amplified. This particular data is from Coriell, and the sequencing details are noted at the bottom. We looked particularly at the categories of SNPs and indels, and specifically comparing recall and precision. And so what you can see here in the data is that they're almost the same. So whether you start with very little and you amplify it, or you have a great deal of DNA to start with and you don't amplify it, the results are nearly identical. The one exception that we've seen is on indel recall. You do see a slight drop, 1 nanogram with amplification, but it's negligible and it varies by sample. We're very conservative in the data that we show, and so this is the most extreme difference that we've seen. So how did we do this? Well, a couple of ways. The first is that unlike older transposomic-based methods that by their very design are unable to capture the ends of the fragments and therefore can't possibly assemble a complete genome with even coverage, we've gone with the classical approach of shearing and ligation. But what's different is that Thermo Fisher Scientific is an enzyme company. We have a vast catalog of reagents that have never been released commercially, and so we went into our bag of tricks. We looked to see which enzymes were most potent, most specific, and most appropriate for sequencing, and we chose those. The reagents and the enzymes that are in these Colibri kits are not in other kits. These are truly unique to Colibri. So what you get from that is a very fast protocol, about an hour and a half, and very sensitive. You don't need very much DNA in order to make this successful. How do we look at this with real samples? Coriell data is nice, but what about real life samples? The data shown in the next few slides was generated by Dr. Carla Tzavova and Dr. Vladimir Benes at EMBL and Mazarek University. These slides are actually from them, and we'd like to thank them personally for sharing this information. What they did was they used the Colibri kits to look at a specific type of sample that is historically very challenging, and that is cell-free DNA, liquid biopsy. And so what they did was they wanted to compare using samples from patients who had been diagnosed with chronic lymphocytic leukemia. They wanted to see the rate of detection of copy number variation between genomic DNA, tumor DNA, solid tumor DNA, and cell-free DNA. Because if the technology, if the Colibri technology is able to detect the same level of CNV with the same accuracy from very small amounts of cell-free DNA as you can from solid tumor or from genomic DNA, that could potentially open up a great deal of additional types of samples with a high confidence in the results. The reason cell-free DNA is so difficult is that it's badly damaged. The average fragment size for cell-free DNA is about 170 nucleotides long. It's also very rare. You don't get very much DNA. And so you cannot possibly risk losing any of it because you're not going to get it back. You didn't have much to start with, and it's not easily recoverable. So what did they find? Well, looking at normal DNA that was obtained from buccal swabs compared to solid tumor DNA from chronic lymphocytic leukemia cells, as well as from cell-free DNA from plasma, 1 nanogram of input of cell-free DNA from plasma, just at 6x coverage, produced the same CNV detection and sensitivity as 10 nanograms of DNA from other types of samples. This is pretty impressive. So they took it one step further. They said, okay, so we can actually use this for real samples, and this is important. What about for more complicated types of studies? So what they did was they used the Colibri kits to look at the next stage of research, and that is really to look at samples that not only had chronic lymphocytic leukemia, but also had pH-negative myeloproliferative neoplasia. And so why this matters is that this is an area where therapeutic development is going on. And so they wanted to find out, again, could they detect the same level of copy number variation with the same sensitivity of detection from people who had donated samples who had received treatment versus samples that had not received treatment. That would be the ultimate test. And so you can see what they found here. What they did was they compared samples that had not received any treatment against samples from people who had received treatment but were in remission following the treatment. And so you've got quite a complex cohort going on here, but this is real life research. This is what we actually work with. And so normal DNA is at the top for control. You can see there aren't any copy number variations there. Next, they looked at the CLL samples. You could detect a copy number variation without any treatment. Then they went into the NPM condition. Again, you could detect it with and without treatment, and cell-free DNA with and without treatment. And the takeaway here is that from very small amounts of sample, they could actually take it all the way through a therapeutic study and make accurate insights into whether that treatment was helping or not. SARS-CoV-2 is an RNA virus, and our gene expression as hosts could be incredibly important, particularly in areas of immunology and our immune response to this virus. And so within RNA sequencing, there's 3 different techniques that are being used to study not only the virus itself, but also host-pathogen interaction. So how do we interact with the virus? What is the impact of infection on our immune system? How do we react? And is there something about our transcriptomes that could indicate susceptibility or predict potential outcomes when infected? These sequencing methods are quite different from each other and provide different types of information. And so what we're going to do in the next few minutes is walk through the different options and the trade-offs between them so that we can think about how to make decisions on which one is appropriate for your particular project goal. At a high level, if we think about the 3 different methods, you receive the most information, the most insights from whole transcriptome sequencing. You get coding and non-coding RNA. You can look at gene expression analysis. You can look at gene fusion. This really is the most complete understanding of the phenotype. In order to get that level of insight and that level of detail, it requires more reads. It's simply the reality of it. So on an Illumina system, generally 60 million reads for a human sample is kind of the minimum in order to get the full information. We've seen labs go up to 100 million or 200 million reads. It's a lot of data to analyze, but you get a lot of insights out of it. Where Colibri can help with this is to make the process of library generation much easier. It's a very rapid protocol. So including ribosomal depletion for human, mouse, or rat, the total workflow time to generate a library is only 6.5 hours. So it's very, very fast. And you start with total RNA in this particular type of sequencing. Now, because this particular sequencing method of whole transcriptome sequencing does require so much data, a lot of people go to mRNA sequencing as a proxy or an initial step. And the reason is that while you don't get non-coding RNA from mRNA sequencing, you do get the information on coding RNA. You can look at gene expression analysis and fusion, and you can do it with fewer reads, typically about 30 million reads for a human sample. The Colibri kit here is, again, very, very fast. It's about a 4.5-hour protocol, and it contains those step-by-step feedback in the form of the tracking dyes. And here you actually start with enriched mRNA. The third category is rather new. This came out in the past year. This is called 3' mRNA sequencing. This sequencing method is very specific to gene expression analysis. So if you're trying to do gene expression profiles of a large cohort of people and you want to see what is the difference among them, and you don't want to spend an awful lot of time digging into complex informatics or large amounts of sequencing, this is the right approach. So 3' mRNA sequencing is interesting because it only requires somewhere between 2 and 5 million reads per sample, not very much. Also, very fast protocol to generate the libraries, about 4.5 hours, but it's also extremely sensitive. You can go all the way down to 100 picograms of total RNA if you wish, all the way up to 500 nanograms. We have a separate seminar. If you're interested in more detail on 3' mRNA sequencing, please reach out to your sales rep, and we're happy to share that with you. So we started with the stranded whole transcriptome sequencing. So let's start there as well and look at that in a little bit more detail. I mentioned that this is very, very fast. Well, it's also very low hands-on time. So to generate a whole transcriptome library, you would deplete your ribosomal RNA, generate your library, about 3.5 hours of hands-on time, and about 6.5 hours of total time. Everything within the Colibri RNA portfolio is highly stranded, very easy. There's a version of the kit available for mRNA sequencing that does not include ribosomal depletion reagents. So if you're not working with human, mouse, or rat, or you really only want poly-A-enriched mRNA, the protocol is even faster. It's about 4.5 hours of total time, and you need somewhere between 1 and 25 nanograms of enriched mRNA to use the kit. Within total RNA sequencing, this kit is designed in a different manner from older library prep technology. And what we've done is create a kit where after you fragment your RNA, you add your helper adapters for Illumina systems. What this means is that you're adding adapters directly to the RNA, not the cDNA. Why does this matter? Well, when you add your adapters directly to RNA and then you complete them into full-length adapters later on during PCR, you actually capture the entire transcriptome. You get your coding and your non-coding, but you also collect small RNA. You get a much richer, robust representation of what's in your sample compared to older technology where the adapters are added to cDNA. There's too many cleanup steps to generate cDNA prior to adapter addition, so we flipped it and said, okay, we want to sequence RNA. Let's put the adapter onto the RNA, then we'll generate our cDNA and put it onto the sequencer. At the other end of the spectrum is the 3' mRNA sequencing kit that we've looked at. So this one's a little bit different compared to Whole-Transcriptome. This gives you gene expression profiles. It does not give you everything. This is a very focused library prep sequencing method. And so what that focus allows you to do is a couple of things. First of all, it allows you to do very short run lengths. So a 1x50 base pair or 1x75 base pair is entirely sufficient to generate 3' mRNA sequencing data that is accurate and robust. And the reason for that is the way that the library technology works is that through a clever use of blocking mechanisms and very specific enzyme choices, it does two things. One, it only captures the 3' end next to the poly A tail, but it also degrades the RNA template in the process of generating the library. What this gives you is the ability to eliminate length normalization. It also tells you that you do not have duplicates that were created by the library prep technology. By having the RNA template dissolve during library preparation, this prevents library prep or technology-induced duplicates. You know that any duplicates you're looking at in your data are true biological duplicates and not artifacts introduced by the library prep technology. And this can really be helpful when comparing gene expression profiles across multiple samples. The other nice thing about 3' mRNA sequencing is that because it only requires somewhere between 2 and 5 million reads, it expands the type of system that you can use out of the Illumina portfolio. So if you require 60 million reads or more in order to do whole transcriptome sequencing, you're pretty much in the NextSeq or NovaSeq or HiSeq category. But if you only need 2 to 5 million reads, you can now go down to a mini-seq if you prefer. This gives you flexibility in timing and cost. If you want to be able to run samples as fast as they come in, run them on a mini-seq. If you want to reduce the cost of sequencing chemistry and you have a lot of samples to run, batch them together and put them onto a NovaSeq. You can actually reduce your sequencing cost per library down as far as $5 per sample by batching these libraries together onto a NovaSeq. This is going to become particularly important as more and more samples start to come into the labs and frankly start to flood into the labs from people who are infected or not infected with SARS-CoV-2 as we look at these gene expression profiles across individuals. So we'd like to point out some data that was generated from Dr. Nicholas Menas at Tulane University, and the following slides all come from Dr. Menas. We thank him for sharing them with us. He used the 3' mRNA sequencing Colibri kit to look at Zika. And Zika is a very good system that was available prior to sample availability for SARS-CoV-2. And previously, sequencing was just simply not affordable. It wasn't within the budget for this particular group. And so they had been using a different technology that you can see on the right here. So the first question is really, how much information is obtained through 3' mRNA sequencing compared to the technology that he was previously using? And so he compared samples from pregnant females that were infected with Zika against males who were infected with Zika to determine the richness of data. Now, this wasn't a gene expression profile study where he truly wanted to see what was the difference between a pregnant female and a male. That's not really the best comparison, as he very openly says. What he wanted to see was about how much data, how much information, what sort of insights would he see. And so he could quickly see there's a big difference. So at a lower cost per sample, the 3' mRNA sequencing technology provided far richer data sets than he previously had been able to receive using older technology. And so he took this a step further, and he took that 3' mRNA sequencing technology, and he's wanted to see what type of insights could be gained from it. And so he was able to tell, just from 2 to 5 million reads, he was able to visualize depressed leukocyte mobilization. He could look at a very specific and extensive rich list of enriched gene sets to look at enhanced metabolic reprogramming. The amount of information provided from 3' mRNA sequencing is so robust from a gene expression profile perspective. This is more than just a technology to be used for grant applications. This is actually providing, at a very low cost, rich data sets for biological insights. So we've talked about 3 different RNA sequencing library prep methods. We've talked about whole-transcriptome sequencing, we've talked about mRNA sequencing, and we've talked about 3' mRNA sequencing, and how to choose between them depending on if we want more data at a higher cost, or if we want gene expression profiling at a lower cost, or somewhere in the middle. But how do you analyze this? Well, if you have an informatician that is available and pipelines in place, fabulous, go with those. If your informatician is currently a bit backlogged or quite frankly, the demand is simply higher than you can keep up with using an informatician, we do have an agreement with Genials, shown here, to analyze data. What Genials has done is to integrate all 3 of the Colibri RNA-Seq library prep kits into their platform, their analysis platform. So you can upload your FASTQs and point and click and generate your annotation. You can look at gene expression differences. You can do your visualization. They actually produce images and reports that are suitable for publication if that's your purpose for the study. Quite a nice system, actually. So what we do is we encourage you, using the email at the bottom, to reach out to them if you'd like to have a demo or to find out about costing on this. This software is designed for biologists. This is not necessarily designed for informaticians, although informaticians might find it completely helpful as well. If you are working on SARS-CoV-2, we encourage you to reach out to them using that email because they will offer the software to you at no cost. There's a lot of different ways to look at this data. We're going to just look at 2 here. Thermo Fisher Scientific doesn't formally endorse or recommend any particular visualization tool or analysis package beyond the Genials software, but you can see examples here. The scientific community has come together to develop quite a few packages specifically to help with this pandemic. So we've looked at nextstrain.org in detail earlier in this conversation. Another option comes from OnRamp. There's a URL there that you can see, and they've done quite a bit of work making their software compatible with many different databases, but also to compare, for example, if you want to look across multiple coronavirus strains for similarities or differences, it's very easy to do with their system. I mentioned at the beginning of this discussion category 3 within the portfolio, which was library quantification. So the library quantification within the Colibri portfolio is qPCR. It has those tracking values. Everything is master mix. The Colibri library quantification kit was designed to be a little bit easier and a little bit faster to perform qPCR quantification of NGS libraries for Illumina systems. And so it doesn't really matter which particular library prep kit you choose, you can quantify it using the Colibri kit. And so we show here some data comparing against another option out there. We show here data comparing against another option in the market for genomes of different GC content and different sizes. You can see different library sizes indicated in the parentheses. So for example, Homo sapien libraries were a little bit shorter in this particular test compared to Pseudomonas, which was a little bit longer. It's essentially the same. So what this means is that you can have the master mixes of the qPCR kit and that visual feedback without throwing out years of data. Essentially the same. It's qPCR. So today we've talked about whole genome sequencing for human and reverse transcribed SARS-CoV-2. We've looked at 3 different sequencing methods within RNA sequencing, again, to sequence the virus, to sequence us, to look at interactions between the 2, and we've touched briefly on library quantification. Samples are available for everything in the portfolio, and they're very easy to request. Simply go to thermofisher.com/colibri-samples. Let us know which one you'd like to try and where we should send it, and we'll send you out a free sample. These samples are all full-size kits, so the library prep reaction samples are 24 reactions. But in addition to sending a sample, we also offer a lot of other help at no cost. If you'd like us to look at bioanalyzer traces or other QC data prior to going into sequencing, sure, we can do that for you. If you need recommendations on how to modify a protocol for your particular sample or experimental goals, we're happy to do that for you as well. Very easy to work with us. With that, I'd like to say thank you. Thank you for joining us in this discussion, but also thank you for the work that you're doing. The work that you're doing or planning to do could be the difference between all of us being able to figure out what normal looks like and having SARS-CoV-2 become a little bit less of a fear and a little bit less of a threat. We have a lot of work to get to a new normal, but the work that you're doing is very important. We thank you. Have a good day. Take care.